Regulation of gene expression. Regulation of gene expression. Regulation of gene expression is the controlling over the function of a gene called regulation of gene expression. The controlling over the function of a gene called regulation of gene expression. This expression, regulation of expression of a gene, we observed in both prokaryotes and eukaryotes. But in prokaryotes, this regulation of gene expression occurs at transcriptional level, only at one point, that is the transcription level. But in eukaryotes, this regulation of gene expression occurs in 4 levels. 1. Transcription level, or during the process of the primary transcript, that is the formation of the hnRNA, heterogeneous nuclear RNA. Processing level, that is during the conversion of the hnRNA into the mRNA. Third one, during the transportation of mRNA to the cytoplasm, during translation level. At any step, this regulation of gene expression may occur in eukaryotes, but in prokaryotes only at one stage, that is the transcription stage. Now in case of the eukaryotes, it can occur at any stage in the 4 steps. One, that is during the transcriptional level, or during the formation of hnRNA from the DNA, or that is called during the formation of the primary transcript, or during the formation of hnRNA. Next one, processing level, during capping, tailing, splicing, hnRNA converted to the functional mRNA, that is called processing level. At that period also it may occur. After processing, functional mRNA is formed. Now this functional mRNA from nucleus that enters into the cytoplasm. At this level also, this regulation of gene expression may occur, or even translation step, it may occur. At any stage in these 4 steps, this regulation of gene expression may occur in eukaryotes. Regulation of gene expression is of 3 types. Regulation of gene expression is of 3 types. Inducible, constitutive, repressible. Regulation of gene expression is of 3 types: inducible, constitutive, and repressible. Inducible gene expression switched on in response to substratum. That is called inducible. Induce, promote. Gene expression switched on in the presence of substratum. That is called inducible. Constitutive, no regulation. That means gene express its character, its nature always. For example, respiratory enzymes. No control. They produce those enzymes, they are produced always. Krebs cycle enzymes, glycolysis enzymes, they are produced always. No regulation. That type of control is called constitutive. Next one, repressible. When the product of gene already present, it's switched off. the gene function is switched off or gene expression is switched off, that is called repressible. So, regulation of gene expression is of three types. Inducible, starts in accordance to substratum present. If substratum is absent, it stops. Start in the presence of substratum, stop in the absence of substratum. Constitutive, always occurs, no regulation. Repressible, if already product is present, product of that gene is present, it stops, switched off. That is repressible. And gene expression, control of gene expression. It may be negative process or it may be positive process. Negative process, switched off because of the presence of the product of the gene. Switched on, it is also the expression of the gene based on product of gene.